Good afternoon, ladies and gentlemen. Welcome to Grand Rounds presented by the Department of Psychiatry and Behavioral Sciences. Thank you for joining us today for the first Grand Rounds of 2025, meaning it's been a quarter of a century since we had Y2K and it's supposed to ruin our computer system. But fortunately, it didn't. Instead, we're actually able to use these computers to join today together for this presentation from the comforts of our homes and offices. I would like to start by introducing myself. My name is Jed Thompson. I'm the Associate Director of Communications for the department. And we have a few logistics to go over here. Let's cover these before we begin. First, we will save some time at the end for questions. So if you have any questions throughout the presentation, please go ahead and write those into Q&A, and I'll get those queued up. We'll save those all for the end. We also have the chat feature enabled, so if you write your questions in the chat, I'll see those too, so either way will work. If you'd like to leave us any feedback, let us know what you thought of the presentation. After the webinar closes, at the end, you'll see a link. It'll pop up. That'll take you to a survey, and you can let us know what you think. And our next presentation actually coming up just next week. Next Friday, the 10th, we'll have Drs. Randall Espinoza and Ryan Kimball joining us. So hopefully you'll all join us again. And one last thing that doesn't have a bullet point there, but just so you all know, we are recording today's presentation. And if you'd like to see it, again, it'll be posted on our YouTube page early next week. So with that, I am going to toss it over to the chair of our department, Dr. Jürgen Unitzer, who will introduce us to today's speaker. Over to you, Jürgen.
All right, thank you, Jed. Happy New Year and welcome to our first Grand Rounds for the year. And today I have the pleasure to introduce our speaker, Dr. Benjamin Srivastava. Ben is an assistant professor in the Division of Substance Use Disorders in the Department of Psychiatry at Columbia. He got his undergraduate degree from the University of Michigan and an MD from the University of Florida. He then completed a general psychiatry residency at WashU in St. Louis and a fellowship in addiction psychiatry and a T32 research fellowship at Columbia, which is where he's on faculty now. Ben's work focuses mostly on alcohol use disorders. And as we all know, this is a chronic illness that is, for many, characterized by frequent relapses. And Ben's research really looks at relapse in, I think, a very, very innovative way. He uses advanced functional imaging methods to look at mechanisms of relapse and recovery in alcohol use disorders. And he was recently awarded an NIAAA K23 Career Development Award to do this research. And today he's going to talk with us about his work to better understand the challenge of protracted withdrawals for individuals with alcohol use disorder, a problem that for many really does contribute to relapse. So with that, please join me in extending a warm welcome to Ben. We really look forward to your presentation.
All right. Thank you so much, Dr. Unitzer. So my talk will be, and thank you everyone for coming. My talk will be on the neural circuitry of negative affect during protracted withdrawal in alcohol use disorder. Just some disclosures. So what will we be talking about today? So we'll be reviewing the background evidence for the proposed circuitry underlying negative affect during protracted withdrawal in alcohol use disorder, present our recent work supporting evidence for this proposed neurocircuit-based model of negative affect during protracted withdrawal in alcohol use disorder, propose plans for interrogating the circuitry and further integrating and validating this model, and propose how fMRI measurements of this circuitry may be utilized as potential biomarkers for personalized medicine and mechanism-based treatment. Okay, so I always like to start off from this quote, and being a WashU trained psychiatrist, Kraepelin was mandatory reading when I was an intern, so I really developed a reverence for descriptive clinical psychopathology, which in these days of epic templates is unfortunately seems like a lost art, but you know, I always keep it alive. So this quote comes from Dr. William Silkworth, who was a psychiatrist in New York a long time ago, and he was the service at Towns Hospital on the Upper West Side, which was a hospital that treated alcoholism and drug addiction. And one of his most famous patients, or perhaps his most famous patient, was Bill Wilson, who was the co-founder of Alcoholics Anonymous. And so after Bill got sober, he asked Silkworth, can you write something for our text, which later became known as Alcoholics Anonymous, or the Big Book. And so this is in a section he wrote called The Doctor's Opinion. So I'll just briefly read it. Men and women drink essentially because they like the effect produced by alcohol. The sensation is so elusive that, while they admit it is injurious, they cannot after a time differentiate the true from the false. To them, their alcoholic life is the only normal one. They are restless, irritable, and discontented unless they can again experience the sense of ease and comfort which comes at once by taking a few drinks which they see others taking with impunity. After they have succumbed to the desire again, as many do, and the phenomenon of craving develops, they pass through the well-known stages of a spree, emerging remorseful, with a firm resolution not to drink again. This is repeated over and over, and unless this person can experience an entire psychic change, there is very little hope of his recovery. Okay, so when I read this, I see a lot of neuroscience, and then I will attempt to convince you why. So, all right. So protracted withdrawal. So I've talked, it's in the title. It's what my K award is about. So we have to define it first. So when we think of withdrawal generally, we first think of acute withdrawal, which is a very well characterized clinical syndrome, as you know, us who have treated these patients know. It begins 6 to maybe 24 hours after the last drink. Hypertension, tachycardia, autonomic instability, anxiety, diaphoresis, insomnia, etc. Then if it's not treated, you start getting the things you don't want: alcoholic hallucinosis, seizures, withdrawal seizures, and then, of course, what you really don't want, delirium tremens. The point is, it's a predictable course. And then when we think about the pathophysiology of acute withdrawal, we think about it on the neurotransmitter level, that alcohol is a GABA-A agonist, and chronic alcohol leads to GABA-A receptor downregulation and NMDA glutamate receptor upregulation, which leads to dysregulation in receptor density and this hyper-excitable state. And so how do you retreat it? How do you treat this? To put it crudely, a GABA deficiency, you give more GABAergic medications. In most cases, and I would argue there's not a reason not to do this, you would use benzodiazepines, though my colleagues at San Francisco General and Massachusetts General find themselves married to phenobarbital, but of course that is a completely separate discussion. Okay, protracted. The point of that is that with acute withdrawal, again, very predictable course, known very sort of well-characterized pathophysiology and a standard treatment algorithm, right? Protracted withdrawal is quite nebulous. Comparatively. So Sally Satel, in her early days when she was at Yale, published a very, very good paper in the American Journal where they rigorously looked at the literature on basically what are psychiatric symptoms for people with substance use disorders after they become abstinent after the acute withdrawal period. And across and within substances, it was very inconsistent, because this idea of protracted withdrawal, we see it clinically, we talk about, people talk about it, patients talk about it, but they said, can we operationalize it? And, the other authors on the paper were Mark Shukett, Tom Costin, and Marion Fishman, who are all very rigorous methodologists, and they said, no, if we are going to include this in the DSM, there's not any way, just based on what we're finding. It is too unreliable, too inconsistent. That's basically why it's not in the DSM. Okay, so one of the words that comes up, or one of the phrases that comes up with protracted withdrawal, is negative affect. So it's a lot of negatively valenced emotions: anxiety, dysphoria, anhedonia. And then one of the one that, at least in the preclinical and clinical literature, people discuss a lot is anxiety during abstinence. And just the presence of anxiety, general or diagnosable anxiety disorders like generalized anxiety, panic disorder. It can definitely affect alcohol use disorder treatment outcomes, and it can increase the risk of relapse. But then there's questions about, well, what is the longitudinal course of anxiety during abstinence? And so the original studies were done by Mark Shukett in the San Diego VA, and basically what he showed is that over the course of about a month of abstinence, people's anxiety returns to baseline. Subsequent work showed that may not always be the case. There have been several papers coming out that one in particular, I think should hopefully be published soon, showing that actually it's a variable course. So then we start to think about mechanism-based hypotheses. So individuals with alcohol use disorder are homeostatically adapted to heavy drinking. Remember Silkworth's quote, their alcoholic life is the only normal one. People with alcohol use disorder are intoxicated. They need to be severe alcohol use disorder, need to be intoxicated. That is their baseline state. And then abstinence causes a deviation from the internal milieu from a homeostatic set point. Homeostasis is disrupted, and then this is where this acute physiologic stress, i.e. withdrawal, comes. Okay, so then we can start to think about what does this look like on a neurocircuit level. So many of you have probably seen this model. It's been around, I think, for about 15 years now, comes from George Koob and Nora Volkov. I like the quote from George Box, all models are wrong, though some are useful, though having NIAAA funding, I have to be careful not to bite the hand that feeds me. But, it's helpful but I think there's some incompleteness to this model. So basically what it is, it's an amalgamation of preclinical and some clinical literature where they conceptualize, Koob and Volkov conceptualize addiction as occurring in 3 phases, and they localize those phases to certain brain structures. So there is the preoccupation anticipation phase that they couple that with defective cognitive control. So a lot of the incentive salience from the ventral striatum and then a dysfunctional prefrontal cortex that's not able to regulate that. And then you have a dysfunctional dorsal striatum that leads to binge drinking. And then after a binge, after repeated binges, one may get this, what we were talking about before, this withdrawal, negative affect, this word George Koob uses, hypercataphia, and they localize that to the extended amygdala, which is generally thought of as including the bed nucleus of the stria terminalis, the central nucleus of the amygdala, and the posterior portion of the shell of the nucleus accumbens. Okay, but again, I don't see it as very integrative. It's basically saying that you have these clinical phenomena that we observe, and then they map sort of neatly onto these areas. When in reality, I think what I think about these systems interacting much more and overlapping. So we have our own model that, and it's kind of a complicated diagram, but I will walk you through it. Everything, I always start with the anterior insula. And so the insula is probably one of the most important cortical regions in substance use disorders. And this starts with Nasser Naqvi's 2007 paper where he showed that patients who had strokes in the insula were readily able to quit smoking. And so this has spurred a whole generation of research. That finding has been replicated, and it's really, really an important area. And so what does the insula do? We learn in medical school this word interoception, that it contains all these visceral and autonomic inputs, and it's this conscious perception of of bodily sensations. And so what does the insula do? So it contains higher order sensory representations of the internal milieu. Remember I said in our hypotheses, or our mechanism-based hypotheses, this deviation from this homeostatic set point causes this acute physiologic stress. See the heart, right? And then there's inputs, again, autonomic inputs, visceral inputs into the anterior insula, and the conscious perception of this withdrawal state occurs in the anterior insula. And we know that the anterior insula has extensive projections to the bed nucleus of the stria terminalis, or the BNST. So what is the BNST? So it's broadly conceptualized as being an area that underlies diffuse or generalized anxiety state. There's a very debatable dichotomy about, whether the amygdala is more acute threats, but I think there is a sufficient amount of evidence to suggest that the BNST is really underlies these more diffuse anxiety states. And so basically what this model says so far is that, okay, there's this acute physiologic stress, the internal milieu is disrupted. This gets consciously perceived by the anterior insula through projections to the bed nucleus of the stria terminalis that coordinates a stress response or withdrawal-related anxiety or protracted withdrawal, right? Acute and protracted withdrawal for that matter. Then we have the dorsolateral prefrontal cortex. So this can potentially put the brakes on this system. So the DLPFC is the area of cognitive control. What is cognitive control? It's the coordination of attention and goal-oriented attention processes and action and representations that can serve goal-directed behavior, right? So maybe staying abstinent, right? And so the DLPFC modulates this system. However, as I'll talk about later, the DLPFC is functionally and structurally impaired in alcohol use disorder. So it may be less able to regulate this system. So again, acute physiologic stress, this is perceived in the anterior insula, projections to the BNST, which create the stress response, and then a deficient cognitive control processes organized by the DLPFC, which is impaired, so there is less regulatory oversight, so to speak, of this circuit. So ideally what happens after enough time is that DLPFC function recovers, so there's improved cognitive control, strengthened connectivity with the anterior insula, so even to the, and then reduced physiologic stress. So reduced physiologic stress, the anterior insula is perceiving less withdrawal, weakened connectivity with the BNST, less anxiety. Even to the extent that there is some protracted withdrawal or still some residual anxiety, then the DLPFC processes organized by the DLPFC can still help regulate it because the DLPFC has recovered. Okay, so this is our model. So what are the current gaps, right? So some questions we can ask are how do function of and connectivity between the AI-BNST and AI-DLPFC change over the course of abstinence? How do these changes relate to specific symptoms of negative affect? How does the DLPFC recovery during abstinence relate to recovery of cognitive control? And so I'm going to present some of our data. So Nasser Naqvi had a data set, and when I was a fellow, he said, do you want to analyze this? And being a fellow and not having my own funding, I said yes. And so this was some interesting data we had. So this was a study Nasser designed. We recruited 38 treatment-seeking participants with alcohol use disorder. They received up to, single cohort, they received up to 12 sessions of cognitive behavioral therapy at STARS, which is our clinical trials unit at Columbia. They underwent fMRI scanning both before and after CBT, and they underwent resting state scanning. And so for those of you who don't know, resting state fMRI is where participants just lie in the scanner, and then you can do a lot with that data. The resting state functional connectivity, which is the signal most people look at, is thought to reflect stable brain states, and changes may index changes related to experience-dependent plasticity. So you could think of it as, okay, remember, as I said, homeostatically adapted to heavy drinking, deviations, maybe a resting state is a good metric for measuring that. Okay, so, and then we had a regulation of craving task, which I'll talk a little bit about later. Again, just for clarification, the reason why there are fewer completers for the resting state is because you have to remove people if there's too much head motion, where that is less of an issue with task data. And there were some other issues why. Okay, and then clinical outcome, percentage of heavy drinking days, greater than 4 for men, greater than 3 for women. This is a standard FDA approved outcome. Okay, so the first question is, okay, well, did CBT work? At least in the single cohort, yes, people drank a lot less. Pretty much everyone reduced their drinking. Okay, which it would have been kind of problematic if that didn't happen, but it did. So, okay. So then we looked at, I had these hypotheses brewing, and we had this resting state data, and I had an understanding of what the resting state signal may potentially mean. So I looked at, okay, what is this what can we get out of this negative affect circuitry, right? So we used a seed for the bed nucleus from Jenny Blackford, who is at Vanderbilt, now at Nebraska. She was the first to segment the human BNST, first in 7T and then later in 3T imaging. The segment is widely available. We used an insula segment from Norm Farb at University of Toronto, and it's very nice because he very meticulously traced sulcal and gyral patterns based on proposed underlying cytoarchitectronics, and you get a very anatomically descriptive insula segmentation. If anyone wants to use it, you can email him. He's very nice and helpful. So the anterior insula is typically divided into the dysgranular region, this pink area, the dorsal anterior region, and the ventral anterior region. And with the dorsal anterior region being thought to be more involved in cognitive cognitive processes, and the ventral more of an emotion area. The ventral region contains level 4 von Economo neurons, is contiguous with the orbitofrontal cortex, and in the substance use disorder literature, along with the OFC and anterior cingulate and nucleus accumbens and medial prefrontal cortex, is thought to be associated with cue-induced cravings. So we looked at baseline resting state functional connectivity between the BNST and the whole insula, just because this is not something that's well characterized. There's one structural fMRI study and some animal studies, but we wanted to get a good idea just at baseline. Are these areas even functionally connected? And so we saw that the BNST is functionally connected with both the dorsal and ventral anterior insula. Okay. All right. So then, and not with any of the middle or posterior regions. Okay. So then we asked, does this change over time? Over the course of CBT rather, and it does not, right? So this is group average data of AI BNST resting state functional connectivity before and after 12 weeks of CBT. And you could say a non-significant increase, but really there's no change, right? But if you start to look at the individual data, participant data, which are these black dots and lines, it looks like there's a regression to the mean phenomenon, right? Like if you're high, you go low. If you're low, you go high. So we thought, okay, well, that's interesting. So, but, still, how do we interpret this? And then remember, people reduced their drinking by a lot. So then it became very straightforward to ask the next question. Do these connectivity changes relate to changes in heavy drinking? Remember, everyone pretty much decreased their drinking. And so what we saw was a pretty robust correlation that the more people reduced their drinking, the more people's connectivity between the anterior insula and BNST reduced, right? And so that, if you think back to our model, that kind of makes sense. There's less drinking, there's less physiologic stress, less interoceptive impact of withdrawal that's perceived in the AI BNST, less functional connectivity. Or you could, and this is the issue with the single cohort, because you can always, the issue is are we taking the effect for the cause? Is there less are the people who are able to, or their brains are able to normalize more effectively, that there's just reduced AI BNST functional connectivity, there's less input from the AI to the BNST, lower anxiety response, lower drinking. Right. So these are preliminary data that more or less fit our model. Right. So then what about the DLPFC? So this was an exploratory analysis. So we basically seeded did a similar analysis before, but we seeded the anterior insula using the same segmentation and looked at changes in functional connectivity with changes in drinking. And this is what we saw. And so basically you see this area of the DLPFC, and it's the 946 area, which I'll talk about in more detail later. But basically, and again, this was an exploratory analysis that we saw, at least at a trend level, that as people reduced their drinking, functional connectivity between the anterior insula and DLPFC increased, right? So it's a lot of, the graph itself is comparing differences to differences. And so it's hard to just look at the map and you may ask why it's negative. But the way if you look at the plot, as people reduce their drinking, their functional connectivity between the AI and DLPFC increased. So maybe it's that there's more DLPFC is recovering. There is with increased connectivity with the anterior insula, the patients may be able to better engage cognitive control processes. Again, consistent with our model. Okay, so we have some data, but what are the current gaps? So how do function of and connectivity between the AI BNST and AI DLPFC change over the course of abstinence? Well, we didn't measure these changes over abstinence, so we still don't know. How did these changes relate to symptoms of negative affect? We didn't measure those to a degree of high fidelity, so that's unclear. And then how does DLPFC recovery during abstinence relate to recovery of cognitive control? Again, we just have a connectivity metric, nothing about cognitive control. So I'm going to talk about that now. So I've said DLPFC a lot. I've said words like 946. So now it's, I think, worth it to take a closer look. So this is a very famous paper from Protracted and Pandya, really almost a tome on the DLPFC. And basically, you can see that it is a very structurally and functionally diverse system, generally thought of as including areas 8, 9, 946, and what they termed as 946. So basically, just a lot of prefrontal cortex, right? But you see the word and people use it inconsistently. And at least for me, when I was starting out, it was very, very confusing as to what people meant. But this area broadly, there's enough in the literature to suggest that in alcohol use disorder, there's impaired function and it does recover with abstinence. As I've talked about, it's a large heterogeneous area of cortex. But then a question one may ask is, well, we always think of DLPFC, cognitive control. 946 is the TMS area that people are interested in. But what about area 8? What is that doing? And so there's different aspects of cognitive control, and could different areas be associated with different aspects, and how does that relate to recovery? Again, just some big picture questions, right? Okay, so one way, there's a lot of ways of measuring cognitive control, again, which is the coordination of attention, action selection, and representations in service of goal-directed behavior. And there's a lot of ways of measuring that. One of those is the Stroop task, the go, no-go task. But the task we use, because Kevin Oxner is in our psychology department, just a train right away, is the cognitive reappraisal. So what is cognitive reappraisal? So it's the reassessment of the meaning of an emotion-evoking stimulus to alter one's response to the stimulus, right? And it's assessed using reappraisal-based tasks. And again, Kevin Oxner developed a lot of these. So basically what the schematic shows, and it's organized a little bit differently than how the tasks are constructed. So there's a stimulus, right? And then prior to the stimulus, there is an instruction that is either look at the stimulus and react naturally, or think about it in a different way, the reappraisal, right? And so this is thought to invoke cognitive control processes. Okay, and it comes from the emotion regulation literature, but it can and has extensively been applied to substance use disorders. So before we get to that, I just want to point out that there have been a lot of neuroimaging studies done, and the findings are remarkably consistent. That you get this very, very consistent pattern of brain activation in the reappraisal tasks. And just to clarify this, these are the contrasts between the reappraisal condition and the just looking naturally condition. But you get this consistent area of the frontal operculum, superior temporal sulcus, posterior parietal lobe, and this area of the DLPFC that's in the posterior middle frontal gyrus, just anterior to the frontal eye fields, that Petrides and Pandya would call area 8AV. And this is sort of an updated map. This is a brain map or brain parcellation from Matt Glasser. That's sort of an updated version of Brodmann based on functional connectivity and some other data that, again, these two peaks from this meta-analysis both correspond to area 8, to this 8AV parcel. So it's a pretty consistent finding, right? Okay, so the regulation of craving test. This is a reappraisal-based test for substance use disorders. It was first designed and published by Hedy Kober in 2010 in smokers, and it's been applied and validated across other substance use disorders. So in this version of the task, participants are shown a highly appetitive stimulus. This is an alcohol task, so either alcohol or highly caloric food, because at that time we were looking at general regulation processes and cravings, not just alcohol specific. But so alcohol or food, and prior to that, an instruction. Look is either just look at the object or look at the stimulus and sort of react naturally to it. And the negative, which is the reappraisal condition, is think about the long-term negative consequences. And then after each cue stimulus pairing, and again, the diagram is just for instructional purposes. Obviously, there was negative alcohol and look food as well. But then after the cue stimulus period, participants are asked, how much do you want this item, i.e. rate your craving. And so the idea is that in the reappraisal condition, craving scores go down. If one thinks about the negative effects of alcohol, he or she will crave less. And so Nasser showed in a preliminary study with non-treatment seekers with participants with alcohol use disorder who are not treatment seeking, that their ability to do this was a little bit impaired, and that and again, consistent with the brain maps I showed you, that the regulation condition, the negative minus look, is associated with this DLPFC area 8AV activity. And so based on this, what Nasser's hypothesis was is, well, in cognitive behavioral therapy, one of the cognitive behavioral therapy for substance use disorder is largely skills-based, and one of the modules has to deal with managing cravings, and one of the key skills is playing the tape through, thinking about the negative consequences. So a mechanism by which CBT may work is that participants may be getting trained to be getting better at doing that, and then that would be associated with increased activity in the DLPFC. Okay, so what did we find? So these are just the task results, not imaging, just the task craving score results. And so what we found was that there's a main effect of instruction, so consistent with previous studies or consistent with the regulation hypothesis that people's craving scores reduced when they went from were reduced in the negative condition as compared to the look condition. There's a main effect of time point. So overall craving scores from before CBT went down craving scores after CBT went down from before CBT. And then there's an interaction effect of Q type and time. So people craved alcohol less after CBT than they did before. What we did not find was an interaction effect of instruction by time. So regulation ability didn't get better with CBT. And we did not find an interaction effect of instruction by Q type by time. So there was not a specific improvement in the ability to regulate alcohol cravings. So at least at a behavioral level, Nasser's initial hypotheses did not hold up. Right. So then we said, well, we have this imaging data. Let's just look at it. Okay. And so first we said, well, did we replicate the previous experiments? And the answer is yes. This is a main effect, or this is the primary contrast of negative minus look, the regulation contrast. As you can see, you get similar areas: frontal operculum, superior temporal sulcus, posterior parietal lobe, and this area 8AV. Again, you can see the peaks of the previous meta-analyses largely line up with this activation map. What we did not, so we basically replicated what's been found before, but then there was no significant effect of instruction by time or instruction by picture by time on DLPFC area 8AV activation. So people did not, there was not an increase in general regulation related brain activation or alcohol cue induced brain activation related activity. So regulation related activity. So essentially, it was a negative paper. Now we published this and there were some other secondary analyses that we did find that are somewhat interesting. But the more I thought about it, that this was a negative finding was the most important part of the paper. That essentially Nasser's hypotheses were not confirmed, and we basically said, okay, well, this is not a mechanism through which CBT works. Moreover, there's not really any changes in brain activity, even though people drank a lot less. Okay, so let's go back to current gaps. So how do function of and connectivity between the AIB and ST and AI DLPFC change over the course of abstinence? We still don't know how these measures change during abstinence. How do these relate to symptoms of negative affect? How they relate to negative affect is unclear. How does DLPFC recovery during abstinence relate to recovery of cognitive control? We did not show that DLPFC activity changed over time. And we have not demonstrated how cognitive control recovers and how this recovery relates to changes in DLPFC function. Okay, so this is my K award will attempt to answer some of these questions, which was just recently funded. So what will we be doing? We will be investigating mechanisms of negative affect during early and protracted abstinence and alcohol use disorder. We'll recruit 40 participants with alcohol use disorder and 20 healthy controls. We'll keep them on disulfiram. So disulfiram or antabuse is a medication used in the treatment of alcohol use disorder. It's been used since 1951. When alcohol is metabolized, it's metabolized by alcohol dehydrogenase into acetaldehyde, which is then metabolized by aldehyde dehydrogenase into acetate, which is excreted. Acetaldehyde, when it builds up, is a very toxic compound, produces the hangover type feeling, and so and disulfiram blocks acetaldehyde dehydrogenase. So if one drinks on disulfiram, one will get a buildup of acetaldehyde and get very, very sick. So as we clinicians know, people have to be motivated to take it, and compliance is the biggest issue. issue. But in a clinic, and I had to tell the NIAAA people this, that I'm not using it as a form of treatment. It is used to keep people abstinent, because otherwise abstinence is very, very difficult to study. But we've shown in some preliminary data that if we pay people to take it, we do remote monitoring, we remind them of what will happen if they drink on it, we can get pretty good compliance, at least in a research setting, for the time that we're monitoring them. So, and then the other advantage is there's no proven, and I say the word proven because someone will always ask about this, CNS therapeutic effect. Yes, there is a large expression of acetaldehyde in the central nervous system. Yes, disulfiram is known to inhibit dopamine beta hydroxylase, which catalyzes the committed step in the biosynthesis of norepinephrine from dopamine, but it was tested quite extensively as a treatment for cocaine use disorder, and those trials were largely negative. And then there was a selective dopamine beta antagonist inhibitor, napikistat. That was a multi-center trial. That's negative. You can look up the results on clinicaltrials.gov. So, I would argue nothing has been definitively proven that there is a definite CNS mechanism of therapeutic effect of disulfiram, as opposed to naltrexone or acamprosate, where they're hypothesized to work based on CNS mechanisms. Okay, we'll get measurements of negative affect. NIAAA put out an RDoc-like matrix with things like the Beck Anxiety and Depression Inventories, the Fawcett-Clarke Pleasure Scale, things that measure all these negative valence emotions. Okay, so what will the study look like? So participants will be induced onto inducted onto disulfiram 24 hours after their last drink. Patients with at risk for severe withdrawal will be excluded. In our clinical trials, we're generally pretty good about doing that and haven't had too many issues. And then to avoid any potential confound of an acute withdrawal picture, like obviously a subclinical one, they will be scanned 1 week after induction onto disulfiram, and then they will be scanned 3 months after, and so that will allow us to really see these changes. And they'll get resting state, so we can look more granularly at the to see if our hypotheses hold up. And then they'll also undergo a task. One of the problems with resting state is it's very, very non-specific, and it's very, very difficult to make causal inferences. And so the question would be, well, is there, and with task, on the other hand, task is a time lock stimulus is associated with a change in bold activity, so you can better make causal inferences. And so the question would be, is there a task that is associated with activity in the BNST and anterior insula? And there is. So this is from Jenny Blackford's group. So this is a very well characterized task. There's lots of different varieties of it, but basically participants are shown in one of 2 images, either a threatening face or a neutral face. And prior to that, they're shown either a cue that will say, okay, you are going to see a neutral face or a threatening face or an unpredictable cue. And the contrast of interest is the unpredictable cue minus the predictable threatening cue, or cue face pairing. And that is very reliably associated with anterior insula BNST activity. So that will add a layer of specificity. And then, so I already talked about that. We'll also get, because there's not a behavioral readout, which would be more ideal, like if the participant were doing something. So we'll have electrodermal activity, skin conductance, which is an autonomic measure of stress and anxiety. And importantly, it's been shown to correlate with AI-BNST evoked activity during unpredictable threat, that unpredictable threat contrast. Okay, so that's the K award. So how do function of and connectivity between the AI-BNST and AI-DLPFC change over the course of abstinence? So the K will help to answer this abstinence question. How do these changes relate to specific symptoms of negative affect? The K will attempt to answer that. And then how does DLPFC recovery during abstinence relate to recovery of cognitive control? So we have not shown that DLPFC changes over time, let alone abstinence. Activity changes over time. We'll be able to get connectivity changes with the K award, or at least be able to measure that, but there's nothing about function. There's not like a DLPFC task. And we have not demonstrated how cognitive control recovers and how this recovery relates to change in DLPFC function. So I'm going to show this slide that I showed before. Again, we did not find, we replicated the primary activation maps, these reappraisal maps, but we did not find any changes in regulation-related brain activity. So then the question is why? And I thought about this for a while, and there's the standard answers like, okay, it was a small sample size, there's not a comparison group, there's a floor effect, and all of these may be true to some extent, but I always had the sense that it was more than that. So the question is are there other processes involved in recovery? So CBT's therapeutic mechanism may not involve the core skill of thinking through the negative consequences to alleviate craving. So I stumbled on this paper from 2017 from Mark Galanter at NYU teamed up with some neuroimaging people. And he actually told me about this at the 2018 AAAP meeting, but then I had forgotten until recently. And so I stumbled on it, and it was really eye-opening. So what they did was they recruited 20 members of Alcoholics Anonymous, with an average sobriety length of 14 years. And these members had to have a spiritual experience, which I'll talk a little bit about. They had to go to meetings regularly. So they were active in AA. And he had them undergo this task, which if you look at it, it's a reappraisal task. Participants are given an instruction. The design is a little bit different. It's a mixed design rather than an event-related design, but it's very similarly structured. But they only viewed alcohol cues. So it was looking at alcohol-specific regulation. And then the instructions were, rather than being shown a negative instruction or think about the negative consequences, they were asked to read AA prayers, the Serenity and the Third Step prayer, and then think about the meaning of them, and then see the image, and then rate their cravings. So a little bit of a different instruction, and then a passive condition, just look at the image and react, and then an unrelated news clip. And I'll get to the results later, but just appreciate very, very similarly designed task, though the difference is the rather than having to think about the negative consequences, they had them reading a prayer and then rate their alcohol cravings. So just a little bit about Alcoholics Anonymous, or AA. So it's a mutual support group founded in 1935 by Bill Wilson, who was a New York stockbroker, and Dr. Bob Smith, who was a colorectal surgeon in Akron, Ohio. And it's premised on the idea of one alcoholic helping another. And at least in its most pure sense is the alcoholic helps another by taking them through the 12 steps, which are a spiritually oriented set of actions taken with the goal of achieving abstinence from alcohol. And so, I heard a lot when I was in training, still hear a lot now. It seems to be the one thing that unites dynamically oriented and so-called biologically oriented psychiatrists, is that you always hear AA doesn't work, it's outdated, you shouldn't tell people to go to church basements, it's a waste of time. And so, I'd hear all of that, but then I'd see patients where that was not true. And so, being a WashU trained psychiatrist, the first question I ask is not how does it make me feel, but what do the data show, right? And so, interestingly, there was a Cochrane review that came out in March 2020, so it got overshadowed by some other stuff, but that showed that, at least in terms of abstinence-related outcomes, 12-step facilitation, which is basically a manualized version of AA, by far outperforms other forms of psychosocial treatment, CBT and motivational enhancement therapy. And the people who did this analysis were John Kelly and Keith Humphreys, who I think are two of the best methodologists in all of psychiatry. I didn't even know they did 12-step research till I read this. So, I think I can trust what they say. So basically, you do see a signal in the literature. And then the other thing is AA is free, right? So yes, you may need to pay money to go to a 12-step based treatment center or do proper 12-step facilitation therapy. But then, it does save a lot of money. And, we spend a quarter of our GDP on healthcare. So that's something I'm always thinking about. And then the other argument that people sort of tirelessly make is, well, it was looking at 12-step facilitation, not AA. And yeah, well, it's not ethical to randomize people and tell them to go to AA versus do something else. So that's why there's 12-step facilitation. And the goal of 12-step is to get people to engage in AA anyways, which it does. So that's sort of that I would consider a very spurious argument. So the signal is AA works, right? And then when you look at mediators of AA's effectiveness, the question of spirituality spirituality comes up, and at least in part, it seems to have some effect, right? Mostly it's the group, but spirituality there is a signal for spirituality. And self-reported spiritual awakening was associated with long-term abstinence and reduced craving, and this may be especially true for more severe patients with alcohol use disorder. All right, and so going to prayer, because remember that was in the task, it's the 11th step of the 12 steps. So what is the 11th step? Sought through prayer and meditation to improve our conscious contact with God as we understood him, praying only for knowledge of his will for us and the power to carry it out. So prayer expands perspective, thus improving emotion regulation. So it could be a form of reappraisal, right? It reduces alcohol consumption in healthy adults. And this was the finding from Galanter's study. They still craved alcohol. These are people with long-term sobriety. They still craved alcohol when looking at the images, but then it reduced when they were asked to read prayers and think about what the prayers meant to them, right? So again, nothing about changing your interpretation or opinion or views on the stimulus itself. It's about changing themselves, right? And so so that was really interesting, just the behavioral findings. Then the brain imaging. Okay, so this was their primary regulation-related brain activation map. And I had to do a double take when I saw this. I actually couldn't believe it, right? Just looking at this map when compared to previous, remember I spent a lot of time talking about how consistent these regulation-related brain activation findings are, right? This area 8AV. In Galanter's study, it's in 946. This is where we saw the signal in our resting state data. This is the TMS area. This is more the area when people talk about goal-directed behavior, this is that area, right? And so this was really, really mind-blowing. And then when you map them both on Glasser's atlas, you can see that they are two very, very distinct areas of DLPFC. And so craving regulation is associated with 946 in long-term sober participants, compared with in reappraisal tests, it's associated with 8AV. So that was really, really interesting to me. And I said, okay, I have to write an R21 to look at this on, to test this, right? So we can go back to cognitive reappraisal. And what does reappraisal mean? So it's the reassessment of the meaning of an emotion-evoking stimulus to alter one's response to the stimulus. And reappraisal can further be broken down into reinterpretation. So mentally changing the meanings, actions, context, and or outcomes of the emotion eliciting stimulus, i.e. thinking about the negative consequences of an alcohol picture. It changes the context and meaning of the stimulus. Reappraisal can also be further broken down into distancing. And what is distancing? It's appraising a stimulus as an objective and partial observer, so the stimulus is no longer as important as before. The key point is it changes the viewpoint of the observer to make the stimulus less impactful. So in some work done on this, largely from Kevin Oxner's group, distancing but not reinterpretation has been shown to reduce negative emotion beyond the effects of habituation. So over time, people's craving scores will go down with regulation, but there's a greater magnitude that shows that you can better train people to engage in distancing than reinterpretation. And effects of distancing-based training appear to be more durable than reinterpretation-based training. So how do we put this all together? So this is the serenity prayer. God grant me the serenity to accept the things I cannot change, courage to change the things I can, and wisdom to know the difference. So it's deeply embedded in AA practices and said at meetings worldwide. And Bill W. even said, never had we seen so much AA in so few words. And central is the message of acceptance, a state of mind characterized by relaxation and freedom from strain and conflict. Thus, prayer in AA may represent a reappraisal strategy based on distancing. It achieves equanimity. It's an emotional detachment from the alcohol stimulus by shifting one's mindset. Again, not thinking about the negative consequences, but changing one's perspective. Again, going back to acceptance, bringing in equanimity. And then repetition is the key to success as a strategy, right? And so again, the interesting thing about those meta-analyses is that they included both reinterpretation and distancing tests, and that you may only get activity in 946 with distancing paradigms after training. right? And then so conversely, in the negative condition in the regulation of craving uses reinterpretation-based processes, which could be like playing the tape through at CBT, the unmanageability and the first step in Alcoholics Anonymous, which I'll talk about in a little bit. And it doesn't really improve with training, and it's associated with area 8AV of the DLPFC. And I really like this quote: AA's 12 steps are a group of principles, spiritual in their nature, which when practiced as a way of life, can expel the obsession to drink and enable the sufferer to become happily and usefully whole. And I like the words expel, practice as a way of life, expel the obsession, like very deliberate. And again, personally, I see a lot of neuroscience there. Okay, so this is an R21. It got good comments, but I need to resubmit it, as is usually the case. So what we'll plan on doing is compare evoked activity and the task contrast that may measure reinterpretation versus distancing based craving regulation in AA. And we'll get resting state functional connectivity. The reinterpretation condition will have them read step 1. We admitted we were powerless over alcohol, that our lives have become unmanageable, because this is basically when someone works step 1, it's basically what are all the bad things that happened when you drank, and could you control your drinking, right? And then the serenity prayer. And then we'll recruit 30 participants in early treatment in a 12-step based treatment, and 30 participants with long-term sobriety greater than 1 year. And these are the DSM cutoffs for early and sustained remission. So it is, that just demonstrates a more valid way of recruitment. So why am I doing this? So it's versus something more generalizable. Well, they have similar methods of staying sober, right? So people in 12-step based treatment are learning the 12 steps, they're learning the mechanics, the vocabulary, etc. And then people with long-term recovery are actually using them. And then you can test with existing paradigms, like, we have the, we can remodel Galanter's test. We can include the first step as a reinterpretation condition. And then again, if you try and get people who haven't been sober, who have been sober through non-AA measures, it's really difficult to interpret what signal you get. Do they not participate in the task because they don't like AA, or does it just work beyond the effects of AA? It's really hard to determine. And also recruitment advantages. So I've been in touch, and they have agreed to collaborate with us. High Watch Recovery Center in Connecticut, which was the first 12-step based treatment center. Marty Mann, who was the first woman to get sober in AA, was involved in the founding. And then we'll recruit AA members from the community, and then both participants will undergo fMRI once. Okay, and this is the task we're using. So we'll have, as I've said, we're using the first step as a reinterpretation condition. Again, instructing them to focus on the unmanageability, because that's all about the bad things alcohol has done. The serenity prayer for distancing, and then a neutral stimulus. And then there's, in most alcohol cue reactivity paradigms, and also in the regulation of craving paradigms, people use static images, but there's been a push in cognitive neuroscience to use naturalistic stimuli because of ecological validity. So with dynamic stimuli in social settings, it's more like real life. Also, interestingly, there's a greater evoked bold response than in static images, and alcohol advertisements, which we will use, are associated with craving and relapse. So I'll just show you a quick example. With Notorious B.I.G. in the background. Anyway, there's another one. Okay, so much more engaging than a static stimulus. And so I'm going to conclude with taking this on another level. So coming from the WashU imaging world, my first introduction was in brain networks. And so what are brain networks? So basically, the concept has existed for hundreds of years, but recently what a lot of people have been doing is using resting state data, and based on changes in connectivity and very sophisticated algorithms, coming up with distributed sets of canonical brain networks that roughly match into task activation patterns, right? And so a question one may ask is, okay, we have these areas, do they map onto brain networks? right? And so when you look at it, our anterior insula segmentation and 946 map onto nodes of their nodes of the cingulo-opercular network. What is the cingulo-opercular network? So it was defined by Nico Dozenbach in 2006-2007 as being responsible for the initiation and maintenance of and control over goal-directed behavior. Basically, he ran a bunch of tasks, and these were the areas that were associated with activity during task maintenance, right? And then 8AV, interestingly, is maps onto a segment of the default mode network. And the default mode network, what is the default mode network? So defined, initially described in 1997 and further validated by Mark Rakel in 2001 with PET imaging. It's basically the areas active when the brain is not engaged in a task or task negative. What is the function of the default mode network? There are many, but monitoring internal states or self-aware or self-directed processes. The interesting thing about these two networks is that they're anti-correlated. So basically, you can look at all the nodes in one network and compare them with all the nodes in the rest of the network, and you get these correlation matrices. And the two most anti-correlated networks are the default mode and cingulo-opercular networks. So basically, the story is in that when the brain is not doing anything, it's in its default state. It's just kind of, it's engaged. It's a very metabolically expensive state. There's a lot going on, but then when it goes into a more goal-directed state, the default mode network backs off. Activity and connectivity in the cingulo-opercular network comes online, and that is responsible for maintaining that goal-directed behavior. So I stayed away from networks for a while because you can theorize a lot about them. There's inconsistency in how they're defined. One of the issues is that there's this debate of, do you need to scan people for a lot longer so you can get these individualized connectivity profiles? This map I'm showing you here is from 120 subjects, but on an individual level, you may see deviations in functional network representation. However, Katarina Grattan a few years ago looked at 10 highly sampled subjects from the Midnight Scan Club, which was the first individualized data set, and she showed that there's good convergence or good consensus that in this DLPFC 946 area as represented by the cingulo-opercular network and this 8AV area as represented by the default mode network. So it's probably not as big of a deal, but it can still be an issue. And then again, is this a better description? And until recently, I said, I don't know. But then I ran into Tim Lauman, who's also the brainchild behind a lot of this. And what he said is, well, we have this paper coming out in Nature, of course, that you may take a look at. And so, this really said, okay, I need to look at networks. So what they did, I'm just going to briefly go over their paper. I would recommend everyone read it. As you see, these blue and orange areas are the somatomotor networks on the motor and primary sensory cortex, primary motor, and then when they looked at task activations, they saw that it only activated a portion, and that in between you had this other. When they looked at resting state networks, they had this other network that they weren't totally sure what it did, and that this other network, which they called the somatocognitive action network, most strongly correlated with the cingulo-opercular network and weakly correlated with the default mode network. And so they looked at Penfield's homunculus, which shows just a distribution of motor function, and they said these regions are inter-effector regions that govern whole body movements. They have task data to show this, and that they're more in service of implementing goal-directed behavior that is organized by the cognitive control areas, i.e. the cingulo-opercular network 946 of the DLPFC. And then I got to thinking, well, this is a mechanism how for distancing-based regulation leads to avoidance of alcohol in settings of alcohol cravings, right? Because we don't have any, who cares what people are doing in the scanner? What about in real life when people are craving, do they go drink or not? And so because distancing is organized by the cingulo-opercular network, which is functionally connected to the somatocognitive action network, that may be a process by which people with long-term sobriety engaged in distancing are able to avoid relapse. Whereas processes involving reinterpretation or unpracticed distancing use circuitry involved in the default mode network, which is not connected to the the scan network, and then they are unable to avoid or less able to avoid drinking using reinterpretation-based strategies. Okay, so what does the R01 look like? I realize I'm almost out of time, so we'll go through this. So examine changes both in negative affect and cognitive control, circuit function, and connectivity during abstinence in AA and 12-step facilitation. We'll use a supervised disulfiram protocol. We'll get measurements of, and I think that's necessary again because you do have attrition with AA. Remember, it was 25% in the project match data. We'll get measurements of negative affect, multimodal imaging design. We'll get precision functional mapping to look at all the connectivity motifs, a threat anticipation task, electrodermal activity, reappraisal tasks with reinterpretation and distancing conditions. We do need a healthy volunteer group to scan longitudinally. And then also just to be sure that there's no CNS effect of disulfiram, we can randomize healthy controls to disulfiram and placebo and scan them. And then once validated, we can probe other interventions that may help 12-step facilitate and help improve attrition and effect sizes in 12-step facilitation, whether that's psychotherapeutic, psychopharmacologic, or neuromodulatory. And I think the most obvious would be neuromodulatory, a randomized controlled trial of TMS versus sham in patients with alcohol use disorder undergoing 12-step facilitation. Despite efficacy for abstinence compared with other treatments, effect sizes are small, there's a lot of dropout. So the question is okay, well, how can we improve attrition? And so there's some preliminary evidence for the DLPFC 946 as a stimulation site in alcohol use disorder. It's obviously the depression area. Then the question that's come up is, well, what about individualized targeting? And this was first conceptualized in Stanford Saint protocol, where they seeded the subgenual anterior cingulate and looked at the most anti-correlated area, which is a 946 area, and that was their stimulation site. And then there were some issues when they did this because you get a lot of signal dropout. Those issues can be circumvented with multi-echo imaging and some other sophisticated techniques. And then you could select on a network level, on an individual network level, the stimulation site of the cingulo-opercular 946 parcel with the anterior insula parcel. Okay, so that is for the future. Obviously, science is a team sport. This was not done just by me, definitely not. My mentors on my K are highlighted in black, and happy to take any questions. Thank you.
Well, thank you as well. We actually don't have any open questions in the Q&A right now, and unless anybody wants to hit enter and send one over right now, I think we'll go ahead and wrap up because we are at 1:01 already. So you actually got it in just in perfect amount of time. All right, so yeah, no last second questions there. So Dr. Srivastava, thank you very much for your time and for sharing your knowledge with us. Much appreciated. Jürgen, do you have anything you want to add before we sign off? All right, well, everyone, thanks for joining us. And please join us again next week, same time, different topic. See you then. Have a good weekend. Bye.